Hello students, welcome to Drishti IAS. My name is Saloni Nandkyolier, and in this session we are going to talk about the deadly Ebola virus outbreak that is being reported in Africa and has now been declared a global emergency. So very recently the World Health Organization has declared the Ebola virus outbreak to be a public health emergency of international concern. So this outbreak currently is being seen in the Democratic Republic of Congo and Uganda. Specifically in the eastern district over here, eastern province Ituri and Uganda, we see this outbreak of Ebola virus, and this has been declared as a public health emergency of international concern, which means that the situation is serious but not as serious as a pandemic like COVID-19. Why? We'll see the reasons later, but for now we need to understand that this has been declared as a public health emergency of international concern. Then when we talk about the cases, so some 300 cases have been suspected and 88 deaths have been reported so far. And this virus or this outbreak is being caused by a virus that is the Bundibugyo virus, which is a rare Ebola variant. Now what is this? When we talk about the Ebola outbreak, it is caused by mainly 6 types of strains or 6 species of virus. Okay, 3 of those 6 are the most important ones or the most common ones, because most of the outbreaks that we see of Ebola happen because of those 3 viruses. So they are, number 1, your Ebola virus, number 2, Sudan virus, and number 3 is your Bundibugyo virus. So the current outbreak has been caused by this virus, the Bundibugyo virus. And why is it rare? Because out of some 20 outbreaks of Ebola that have happened so far, only 3 of those 20 have happened because of this virus, because of this strain. Okay, so this could actually be a question in the prelims examination, that the current outbreak of Ebola that we see in Africa is being caused by the Ebola virus. So this could easily confuse you, because it is not being caused by the Ebola virus but by the Bundibugyo virus, okay, which is a rare Ebola variant. So WHO has said that this is a global health emergency, it's a public health international concern and everything, but it is not a pandemic like COVID-19, because COVID-19 was airborne. So the reach of Ebola is not as much when we talk about airborne diseases. So they can easily be spread. They're just like flu, cough, cold. They can actually be transmitted very easily from one person to another. So the reach was very high. It was a global disease. It was being spread so quickly, and we wanted to contain the disease. That's why, or contain the virus. That's why, we were taking very stringent steps like closing the international borders, quarantine and everything, which is not the case with Ebola virus, because Ebola is not airborne. Ebola is not airborne. It is actually transmitted through bodily fluids. Bodily fluids like blood, vomit, semen, etc. But because it is not airborne, so the transmission is not as high as it was in COVID-19. So it's not a pandemic yet, okay? But we still need to prevent things because the matters are getting serious in Africa. So the things have to be monitored. Borders have to be monitored. Patients have to be monitored. But closure does not need to happen, okay? Next, if we talk about this disease, the details of the Ebola virus disease. So earlier this was actually called Ebola hemorrhagic fever, and this is a severe and often fatal infectious illness affecting humans and other primates. So it affects humans also and other primates also, like monkeys and gorillas, chimpanzees, etc. So it affects humans, it affects primates, and it is often fatal. The death rate over here is high. In fact, in a couple of cases or couple of outbreaks that we have seen in the past, the death rate has gone as high as 90%. 90%, meaning almost everybody who was contracting the disease was actually dying. So it has gone as high as 90% also, and right now it is somewhere around 50%. Okay? Then when we talk about this virus, Ebola virus, this belongs to the Ortho-Ebola virus genus. Ortho-Ebola virus genus, and the family is Filoviridae. Okay? And I already told you that 6 species of this ortho-Ebola virus genus actually cause Ebola outbreak. 3 out of those 6 are very important, like the Ebola virus. It's like the most common one. This strain, Ebola virus, is actually the most common virus that causes almost all the outbreaks. Then we have Sudan virus, and then we have the Bundibugyo virus, which is rare because only 3 of 20 cases have reported this virus, and the current outbreak is because of this virus. And why is this virus so important? Because we do not have any approved vaccine or therapeutics to actually cure this particular virus. That's why it is more like it's deadlier in comparison to the other strains that we have. Okay? Then when we talk about its origin, like from where does it get this name? So for the first time it was reported, or it was identified in 1976 simultaneously at 2 places, because the outbreak was simultaneous at DRC, Democratic Republic of Congo, Yambugo, DRC, near the Ebola River. Ebola River, hence the name. And Zara in South Sudan. So these 2 outbreaks happened simultaneously, and because this virus was discovered near Ebola River, hence it got its name. That's the Ebola virus. Okay. Then when we talk about its transmission, so this is a zoonotic disease. Zoonotic disease meaning that it originates in animals, and from animals it actually affects humans. Okay. So we see animal to human transmission, and here fruit bats. Fruit bats of the Pteropodidae family are actually the natural hosts of these viruses, of the Ebola virus. So from fruit bats or from these animals, it can actually spread to other animals also, like primates that we saw, monkeys and apes. Then it can also spread to porcupines. It can also spread to forest antelopes. So animal to animal transmission can also happen. Animal to human transmission can also happen, either through the direct consumption of the infected animal by the humans, or through bodily fluids like your blood, vomit, fecal matter, etc. And then human to human transmission also takes place. Human to human transmission again through bodily fluids. There could be vomit, semen, stool, blood, or contaminated needles, or even by sharing contaminated bedding, contaminated clothing, wherein any strain of that blood could be there. So through an infected person, it can also affect other healthy people. It's a zoonotic disease. It originates, or the natural hosts that we see are fruit bats. Animal to animal transmission also happens. Infected animals can transmit this to other healthy animals. Then animal to human transmission happens, and human to human transmission also takes place. Okay? And when we talk about the mechanism that it follows inside the cells, this virus, then the first step over here is that the virus that you see, this is the virus, okay, this is the Ebola virus. It has certain protein. It has certain protein on the surface, the glycoprotein. Glycoprotein. Now this protein is going to identify or target some specific types of cells, and once the target is identified, the virus is going to enter the cell, okay, enter the healthy cell. And once it enters the healthy cell, it is going to release its genetic material. And the genetic material over here for Ebola virus is your single-stranded RNA. Single-stranded RNA. So virus enters the normal cell, healthy cell. There it is going to release its genetic material, and now this virus, this genetic material is actually going to take over the cell's machinery. It is going to hijack the cell. It will take over the cell's machinery and in turn start replicating itself. So the genetic material will start producing more copies of itself. More RNA, more virus is going to be created over here. So ideally what would have happened if everything would have been working fine, then the cell machinery would actually develop proteins that the body needs. But now because the cell machinery has been hijacked by the Ebola virus, so it is not going to function properly. Instead, this is going to, like this genetic material is going to replicate itself. It will keep on producing more and more viruses. And then finally, these viruses are going to be released again in the body. And then they are going to affect other cells also. And exactly same procedure is going to follow. Everywhere they are going to create copies of themselves, and they'll start affecting all the other, in proper or healthy cells also. So that's why the transmission happens very quickly. Transmission happens, and then the fatality we also saw, it's quite high. It is right now it is as high as 50%. Okay. So when we talk about the symptoms, the incubation period over here is some 2 to 21 days. It takes some 2 to 21 days for the symptoms to appear. The initial symptoms are basic, like you would have fever and nausea, vomiting or headache or severe muscle pain. And then advanced could be diarrhea, rash, and then impaired kidney and liver functions could also be reported. And internal and external bleeding can also happen. Okay, internal and external bleeding can also happen in certain cases. The death rate, I already told you, it is high, 50%. And in some of the cases, in the past outbreaks, we have seen the range from 25 to 90%, depending on the virus strain and the medical response. Correct? The current one is Bundibugyo virus. We do not have any approved vaccination, any approved therapeutics to deal with that. And how does the diagnosis take place? So it's diagnosed by mainly two types of tests. We have RT-PCR and ELISA. RT-PCR, reverse transcription polymerase chain reaction, and ELISA, enzyme-linked immunosorbent assay. Now what happens in RT-PCR? I told you that the virus is entering the body and it is releasing what? It is releasing its genetic material. It is releasing its DNA. So what do we do in this? There is actually a chain reaction. Now, and especially helpful in the initial stages. Because in the initial stages or in the initial days when the virus has just entered the body, they are going to be few in number, right? They are going to be few in number, and it gets difficult to detect these viruses because they are not, as abundant in number as they would be in the later stages. So they are few in number. What do we need to do? We need to create more copies of that, or we need to amplify that so that we can detect it. So because the RNA is very limited in number, in that case the detection becomes difficult. So in this chain reaction, what happens? We create copies. We create copies of DNA and not RNA because it is easier to make copies of DNA. So what do we do? In the first step, we actually convert this RNA to DNA. This is your reverse transcription, right? Because transcription is DNA to RNA. RNA to DNA is your reverse transcription. So first step would be the conversion of RNA to DNA. This is going to be formed, and then this DNA through chain reaction, polymerase chain reaction, it is actually going to replicate itself and create millions and millions of copies, right? So it's like a photocopy machine. Now what will happen? If there was no RNA, if there was no viral genetic material, if the body or if the person was not infected, no copies will be formed because there was no genetic material or there was no RNA. RNA will not convert to DNA, then DNA will not make its copies, and we will not see anything. The test will come out to be negative, right? But if the RNA was present, even though in tiny amounts, if the RNA was present, because it was in tiny amounts, it was difficult to detect it. Now this RNA is going to convert to DNA. We are going to create millions of copies of DNAs. And now if these copies are being created, that means that the virus was present, right? So the test will be positive. So that's why I said that especially in the initial stages, and we saw this in COVID times also, RT-PCR was being used, particularly in the initial stages. There were home tests that were happening, so that if there was anything, if there was a slight strain of virus that was present, this test will be able to detect it because of the chain reaction that it follows, right? Then we have ELISA also. ELISA is actually a color-based test. A color-based test which actually makes use of an enzyme. So if there is any antigen and antibody, okay, so it is going to identify both the antigen and the antibody. Antigen is your virus itself, right? Antigen is the virus, And antibodies are the response that are being generated by your immune system. So if there is the virus and if there are antibodies in either of the case, this ELISA is going to be helpful, because the color, there's a chemical solution that is used. It does not have any color in the beginning, but if there are antigens present or if there are antibodies present, then a certain enzyme is going to be triggered, and then the color of that chemical solution will change. So if the color changes, that means either the antigen or the antibody is present, which means that the virus is present. Virus is present and the person is infected. So these are the two types of tests that are used the most when we talk about diagnosis of the Ebola virus. And once diagnosed, then when we discuss the treatment, so the initial treatment is rehydration with oral or IV fluids, and then this special type of treatment is also used, which is the monoclonal antibody treatment. Monoclonal antibodies are used. What are these monoclonal antibodies? These are not your regular antibodies that are created naturally in the immune system of the body, but these are the antibodies that are made in labs. These are the antibodies that are made in labs, monoclonal antibodies, and they are specially helpful because they they have a very targeted, specific delivery. So unlike the regular antibodies which are going to target different types of viruses, or even at times healthy cells can also get affected. When we talk about these monoclonal antibodies, they are engineered inside the laboratory to actually take care of or to actually fight with the exact type of virus. We identify the concern and then we engineer things in the laboratory, and then we make these antibodies. Then we make these monoclonal antibodies. And because they are so specific and targeted in their delivery, they are not only used in Ebola virus treatment, they are also used in cancer treatments and autoimmune disorders. Because there also we don't want the healthy cells to be affected, and we want a very specific, very targeted delivery. So that's why these monoclonal antibody treatments are given in these cases also. And in the Ebola case also, we have this antibody treatment. Some names have been given over here: Inmazeb and Ibanga. They exist for the Zaire strain or the Ebola strain, the Ebola virus. Okay, but I already told you that we don't have any specific vaccine or therapeutic for the Bundibugyo virus, but we have two vaccines that have been approved for the Ebola virus, that is Ervebo and Zabdino or Wabia. Okay, and for the current outbreak also, as a response mechanism, Ervebo vaccine is being recommended, which is ideally or originally for Ebola virus only. It's not exactly for Bundibugyo virus, but as a response mechanism, this vaccine is also being promoted. Okay, so that was all for this discussion. Let us hope that the situation in Africa does not get any more severe. But this is the situation. It's actually, I hope you were able to understand what Ebola virus is, how is it transmitted from animals to humans. It's a zoonotic disease. How is it spread and how can we actually treat it, right? Now we look at a practice question for prelims. Consider the following statements regarding Ebola virus. 1. Bundibugyo virus is a rare variant of the Ebola virus. 2. Ebola virus is primarily a zoonotic disease. Which of the statements given above is or are correct? A. 1 only, B. 2 only, C. both 1 and 2, or D. neither 1 nor 2. Please attempt this question and provide your answers in the comment section. Thank you for watching.
For more informative content, like, share and subscribe, and do not forget to press the bell icon to get the notifications.